everyone. From today, we are going to talk about the development that happens during the embryonic period. So when we talk about embryonic period, we are going to mainly focus on the events that happens between the 3rd week to the 8th week. So almost till 2 months of pregnancy period, what are the events that happen is what we are going to cover under embryonic period. So our last class was on 3rd week development, where we saw two things, right? One was gastrulation. So with respect to gastrulation, you should know that formation of primitive streak begins the gastrulation. So it is the formation of 3 layers. So we saw the ectoderm, mesoderm, and the endoderm was formed. Then we were also talking about the buccopharyngeal membrane that is formed as from the procaudal plate, and the cloacal membrane that is formed along the connecting stalk. And we saw why the membranes are formed there, right? Then we spoke about notochord. In the notochord, you have to know the primitive node, primitive pit, notochordal process in the notochordal canal. Then there is going to be intercalation with the underlying layers. So it forms a notochordal plate which will flatten. Then it is filled by the mesodermal cells. Again, it forms a definitive notochord. So these were the events that we saw in the last week, right? So now we are going to talk about the very important thing, that is neurulation. So neurulation is going to begin at the 3rd week, approximately around 19th to 20th day, and it is going to get till 28th day, right? So this is neurulation, and along with neurulation, I have also included the development of brain, though it is not a part of 3rd week development. Yes, it is going to begin after the neural tube is formed. So instead of covering it in systemic, I thought, okay, let me combine with neurulation so it becomes easy for you to integrate and study. So without any delay, let's get started. So with respect to neurulation, yes, this is a very beautiful picture that will enable you to understand what happens with neurulation. Yes. So what is happening there? You all know that at the 3rd week, we have got formation of trilaminar germ disc, right? We have got the formation of trilaminar germ disc. So this red color one is what is your ectoderm, and this is your endoderm, right? Now what happens in the ectoderm is, can you make out, he has given something called as the neural plate. Yes. Now the notochord is formed here, right? So here we have got the notochord. Now this notochord is going to send some signals. It is going to send some signals to the above placed ectoderm. Yes, it is going to send some signals to the ectoderm, and because of these continuous signals that is being sent to ectoderm, there is a formation of a structure called as neural plate. Yes, so where this neural plate is located is very good. It is located in the ectoderm. So what is the source of formation of neural plate, or who is going to provoke the ectoderm to form neural plate? This is the signal from the notochord. So signal from the notochord is continuously asking the ectoderm to form a structure called as neural plate. And the cells that are present along the neural plate are called as Neuro Ectoderm. Right, so this region of purple color one is your Neuro Ectoderm, the neural plate, and this neural plate is the one that is going to give rise to neural tube, right? So just remember this alone. So initially I got a plate, then it is going to form a groove, then I get the tube. So this is how the process goes. So just have this picture, let us get into our embryological picture study, and once again I'll put this picture so that you will be able to understand neurulation. Yes, so what happens is, these neuroectodermal cells, right? This neuroectodermal cells will start invaginating the mesodermal layer. Yes, it is going to begin to invaginate. So say suppose this is my normal ectoderm. So this normal ectoderm, I call it as surface ectoderm, and this region, okay, this yellow color one that I'm going to highlight here now, this is my neural plate containing Neuro Ectoderm. Now what is happening to this Neuro Ectoderm is it is going to form a groove-like structure. So it is going to invaginate into the mesoderm, and it will begin to form, a neural groove. Right? It is going to form a neural groove. Now this is the junction between the surface ectoderm and the neuro ectoderm, right? So this yellow color one is the neuro ectoderm and this is the surface ectoderm. And no, there was a point where these two are going to meet, right? So that is called as the flank of neural groove. That is the junction between the neuro ectoderm and the surface ectoderm. So in the junction of these two, appear a group of cells, and that cells are called as crest cells. So we have seen three structures: neuro ectoderm, that is going to give rise to neural groove. So this is my neural groove. And in the junction of the surface ectoderm and the neuro ectoderm, as I am seeing a population of cells, and that population of cells, I call it as crest cells, right? I call it as neural crest cells. Again, a very important group of cells. So we'll see what happens. Yes, so this is a picture, right? So this portion is my neural groove. Yes, this is my neural groove, and this is my surface ectoderm. And can you make out the junction of the surface ectoderm and the neural groove? Yes, it is present with certain cells called as neural crest cells. And this is my notochord. And these are all something called as somites that we will be seeing in the next class, right? So they are all part of the intraembryonic mesoderm. So this is your somite. So when the neurulation begins, you see all these structures. So you have got a neural crest, you have got a notochord, and side you have got some part of intraembryonic mesoderm that is going to form something called as somites, right? So this is how initially it will look, right? So neurulation begins with the formation of neural plate. Then I have got something called as neural groove. So if you take, this is a picture that is seen on the 19th day. Yes, can you make out, this is the ectodermal cells, and here I will be having my neural plate, and slowly it had began to give the neural groove. Yes, can you appreciate in this picture? As the neural groove have very well appreciated here. So this is the fold, right? So what happens, the plate had folded, right? And then it has started to give a groove. So this is called as a neural groove, and side I have got my somites, right? Now let us draw that picture again, the one that I showed you in the first slide. Yes, so this is my surface ectoderm, right? So this is my surface ectoderm, and this is the going neural groove, and in the junction I have a group of cells called as neural crest cells. Now what happens here is, as this process, this folding is going to be a continuous process, right? So this groove is formed as a result of folding. So what happens is, the neural plate on both these sides, so say suppose if the neural plate would have been like this neural plate, as it undergoes some foldings like this, and this, as a result of that folding only, I get this neural groove. So what happens is, every time when this folding, this groove is formed, this surface ectoderm is going to approach each other, right? Just imagine it is continuously invaginating, and it is going to form groove continuously. So this groove is going to form continuously, and slowly it increases in length. So when this groove is formed and it increases in length, what happens is, yes, they are going to approach each other along the midline. So our Langman says this neurulation, when the neural groove is formed, the cells are all moving from the lateral aspect to the medial aspect, right? So when this folding is happening, each of them are going to approach along the medial axis. So continuously the foldings are going to take place, and all these cells are going to approach each other in the midline. Yes, it is very important. Now, once they are approaching towards the midline, at one point they are going to fuse. Just imagine now, initially I got a picture like this. Then the surface ectoderm is approaching now. So what will happen when these cells are going to approach, the surface ectodermal cells are approaching, the groove becomes even more steeper, right? So if you take the difference between the width of the groove, yes, it has been different. So what happens is, these cells are all going to approach each other from the lateral aspect to the medial aspect, and the groove is going to become even more deeper. So at one point, what will happen? These surface ectodermal cells will completely fuse. Yes, they will completely fuse. So when these cells are going to fuse, yes, when these cells are going to fuse, this groove is going to come as a tube-like structure. So they will detach from the surface ectodermal junction and they form a separate tube-like structure. So along with them, the neural crest cells will also come and it is going to stand like this. Yes. So what I want to tell you here is, we'll get back to this slide again. So this is the neural plate and we saw the neural groove. Can you appreciate here? It has formed a tube-like structure and it has completely come out from the underlying epidermal cells, that is the ectodermal cells. So what had happened here is, can you make out, so here if you take, he has given something called as convergence. So what happens is, these cells are going to converge from the lateral aspect to the medial aspect, and that is why the fold had begun. So they begin to fold. These neuroectodermal cells begin to fold. As a result, we get the neural groove. And what happens? Slowly they approach towards the medial aspect. And one point they are going to meet. And when they are going to fuse with each other, this neural groove is going to detach from the underlying ectodermal cells and form a structure called neural tube. When this neural tube is formed, along with them, I also get the neural crest cells standing in the mesoderm alone. Right? Did you get my point? So this is how a neural tube is formed. So this is a well-developed neural tube. And what is more important here is, you have to know two openings. Now, this is the developing embryo, and surrounding with it, I'll be having the amniotic cavity, right? So surrounding, I'll be having the amniotic cavity. So, right. So what will happen is, the neural tube is going to communicate with the amniotic cavity anteriorly by the anterior neuropore and posteriorly by the posterior neuropore. Very, very important. So when the neural tube is formed, as along with them, there are two pores that appear on the anterior and the posterior aspect, or I can say it as the cranial and the caudal aspect. On the cranial side, I have got the anterior neuropore, and on the caudal aspect, I have got the posterior neuropore. Now, why are these pores important is neurulation is marked to be complete when both of these pores are going to close, right? So closure of the pores marks the end of neurulation. So the anterior neuropore will close approximately around the 25th day, and the posterior neuropore will close approximately on the 28th day. Very, very important. Yes. So once this closure of pores had taken place, it marks the end of neurulation, and I can say a neural tube is formed. Yes, neural tube is formed. So to summarize, is what you have to know. The first structure you have to mention is about the neural plate that develops in the ectodermal layer. As a result, I get neuro ectoderm and the surface ectoderm. And these neuro ectodermal cells present in the neural plate will start to form foldings. Okay, so it is going to fold in such a way that the cells are going to converge from the lateral aspect to the medial aspect. This folding is going to form a groove-like structure, and that groove is called as the neural groove. Right? And these convergence, right, the convergence of the neuroectodermal cells happen continuously. At one point, they fuse. As a result of fusion, the neural groove along with the crest cells, they detach from the underlying layer and form a separate structure called as neural tube. So this neural tube, right, it is marked by, so as it keeps growing, this tube is characterized by the presence of anterior neuropore and the posterior neuropore, which has to be closed. That marks the end of neurulation. So anterior neuropore closes at the 25th day and the posterior neuropore closes at the 28th day. Right, both will establish a connection of the embryo with the amniotic cavity. Right, so this is about your neurulation. Now, we have to understand the abnormality. So with respect to neurulation, you have to know about how the brain develops, and you have to know the abnormalities. Very, very, very important. Now, we told that the anterior neuropore has to close and the posterior neuropore has to close on a particular day, right? 25th day, the anterior has to close, and 28th day, the posterior neuropore has to close, and that will mark the end of neurulation. If these pores are not going to close, it leads to serious anomalies. So, if the anterior neuropore is not closed, so failure of anterior neuropore closure will lead on to a condition called anencephaly, absence of cephalic end, anencephaly. So brain is not going to develop, right? So as a result is that no doctors will prescribe abortion. So medical termination of pregnancy will be the ultimate solution for this, right? So anencephaly can happen if the anterior neuropore fails to close approximately on the 25th day. Now, the second thing you all must have heard by now is something called Spina Bifida. Now, Spina Bifida, yes, obviously it is going to be failure of closure of posterior neuropore. Failure of closure of posterior neuropore. If the posterior neuropore fails to close, yes, I get an abnormality, a neural tube defect called spina bifida. So this can happen anywhere on cephalic, caudally, right? But the most common site is the lumbosacral region. This can be a question. Spina bifida is most common at the lumbosacral region, right? So these are all neural tube defects. Now, what is the most suggestion to prevent this neural tube defect is to administer folic acid tablets. So in the first trimester, all the pregnant women are given with folic acid supplementation to avoid neural tube defects, because for neurulation, for the neural crest formation, right, for the neural crest cells and the neural plate, yes, they require folic acid. So folic acid supplements have been given right from the first trimester. Once the pregnancy is confirmed, we start with the folic acid supplement so that neural tube defects are avoided. Right, so failure of closure of anterior neuropore, anencephaly, closure of posterior neuropore is the spina bifida. Right, so what are these neural crest cells? So we saw that some of these cells from the Neuro Ectoderm is going to migrate along with the neural tube. Those are called as neural crest cells. And once the detachment takes place, once this approximation takes place, as the neural crest cells are going to just float, or it is going to be present along the mesodermal layer. So what is the role of neural crest cells? So once the neural crest cells are detached and they float separately along the mesoderm, it's not float, it is going to be there at the mesoderm. It takes two pathways. One is called as the dorsal pathway. The other one is called as your ventral pathway. Now, through the dorsal pathway, it gets to dermis. So D for D, dorsal pathway, dermis, and it forms melanocytes. So sometimes the neural crest cells can get into a dorsal pathway, it can form through the dermis and it forms melanocytes. Or it can get along the ventral pathway. Yes, it is going to get along the ventral pathway. So you have the somates along the ventral aspect, and it is going to give rise to so many structures like your ganglion, your sympathetic nerves, etc. So since it is going to give rise to so many structures, sometimes neural crest cells is referred as the fourth germ layer. Yes, it is referred to as the fourth germ layer. So whenever neurulation is asked, please make sure that you mention about the neural crest cells. What happens to the neural crest cells? Right, so this is about neurulation along with the neural crest cells. Let us quickly get into the CNS, brain development, brain and the spinal cord. Mainly I'm going to focus on the brain. Right, so once the anterior neuropore and the posterior neuropore is going to close, right, neural tube is formed. So this closed neural tube is what is going to give rise to CNS structure. So what happens is, once the neural tube is formed, the cranial aspect of the neural tube is going to give rise to brain. Yes, this is the cranial aspect. The cranial aspect of the neural tube gives rise to brain, and the caudal aspect is going to give rise to spinal cord. Yes, it is going to give rise to spinal cord. So this is the neural tube, and if you take initially, I will have three parts. One is the forebrain part, the midbrain part, and the hindbrain part. So what is this forebrain part? It is referred to as, yes, it is referred to as prosencephalon. This is called as mesencephalon. We would have heard these terms, right? And this is called as rhombencephalon. So once the neural tube is completely closed, yes, the cranial part is going to give rise to the brain and the caudal part gives rise to spinal cord. So what happens is all these are going to appear as vesicles. Yes, some round structures like vesicular structures, and vesicular structure that is present on the topmost, the cranial most part is the prosencephalon, in the middle is the mesencephalon, and in the lower aspect is the 𝗶𝗵𝒂𝒃. So prosencephalon will give rise to forebrain, mesencephalon to the midbrain, hindbrain, is given by 𝗶𝗵𝒂𝒃. So initially you have three brain vesicles, right? So these are called as primary vesicles. These three, pro, meson, rhomben, are called as primary vesicles. Soon what will happen is they give rise to secondary vesicles. Yes, they give rise to secondary vesicles. So what happens is the prosencephalon, will divide into two. Yes, can you make out? One is the telencephalon, another one is diencephalon. Telencephalon and diencephalon. And the mesencephalon is going to exist as mesencephalon only. And similarly, if you take the rhombencephalon, yes, if you take the rhombencephalon, it gives rise to two structures again. One is the metencephalon, another one is myelencephalon. So totally for the development of brain, there are 5 vesicles, right? 2 from the prosencephalon, 2 from the rhombencephalon, 1 from the mesencephalon. So we have to know what are the derivatives of each. So if you take telencephalon, the topmost part, so it gives rise to the cerebral hemispheres. It's going to give rise to cerebral hemispheres. If you take diencephalon, yes, you all know diencephalon is structured closely associated with thalamus, right? So hypothalamus, thalamus, subthalamus, all the thalamic structures are given by diencephalon. Along with that, it also gives rise to pars nervosa, that is your posterior pituitary, along with optic vesicle. So derivatives of diencephalon are very important. So thalamus, hypothalamus, subthalamus. Along with that, it gives rise to pars nervosa and optic stalk or your optic vesicle. So optic vesicle gives rise to the optic nerve and etc. structures. Right? Now, mesencephalon you take, as we saw, it is going to be midbrain derivative. So all these structures are corresponding to midbrain. Your colliculus and all them. Right? Similarly, if you take metencephalon, that is the rhombencephalon, we have got 2 derivatives. First is the metencephalon, and the more caudal part is your myelencephalon. So the metencephalon is going to give rise to cerebellum and the pons, and the myelencephalon is going to give rise to medulla. So you all know these are all parts of hindbrain, right? So pons, medulla, cerebellum, all these are aspects of hindbrain. So cerebellum and pons from the metencephalon, and myelencephalon is going to give rise to medulla oblongata. Now, when these structures are developing, each of these vesicles are developing, there appears a cavity. And each of those cavities is going to give rise to ventricles. For example, if you take the cavity of the rhombencephalon, yes, this portion, this cavity is going to be your 4th ventricle. Cavity that appears in the mesencephalon is your 3rd ventricle. Cavity that appears along the telencephalon, that is the prosencephalon, is going to be your lateral ventricles. So you all know we have got the lateral ventricle, 3rd ventricle and the 4th ventricle. So the cavities appear in all these brain vesicles, and each of them is going to give rise to ventricles, right? So initially we have 3 brain vesicles, primary vesicles, that in turn gives rise to 5 vesicles, right? And we have seen the derivative of each of these 5 vesicles, and that marks the end of development of brain, right? So to summarize, as we saw about the neurulation. So in the neurulation, I have already told you what are the structures you have to know, right? Neural plate, neural groove, right? Neural crest cells. What happens to neural crest cells? Then we saw anterior craniopore and the posterior pore. All these has to close. So we saw 25th day the anterior pore closes, 28th day the posterior pore closes, and once this closure happens, it marks the end of the neurulation, and this closed neural tube is going to give rise to brain and the spinal cord, and we saw how the brain and the spinal cord develops, right? So when I start the next topic, I'll once again revise all these, and then I'll start with the next topic, right? So thank you. See you in the next video. Take care. Bye.